This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 561, recorded on August 16th, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. It's a pleasant summer day here, late summer day, mostly cloudy, 84 Fahrenheit, 29 C, and a good day to do TWiV.
Yeah, it is the last 2 weeks of August. It's kind of over. Yeah. It's amazing how quickly it went here.
School starts soon.
26 C and cloudy. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. I'm a little sad that you were talking about how school starts soon because you're reminding me of my syllabus that I should be working on.
Oops.
Well, yeah, you got the whole weekend to work on it.
I have the weekend and next week.
That's plenty of time. Plenty of time. I mean, presumably you have a previous year's syllabus you can modify, right?
I do, yes. It's just been nice not thinking about it recently. And it's also gray and 27 here.
Yeah, same as here. So quiet this week. This week, driving in, there was no traffic. On any day, it was like an hour drive. It's just been amazing. I wish it were like that every day.
You have an hour commute?
When it's good.
Wow.
It's 38 miles, so an hour. I could do it in 50 minutes if there's zero traffic. Like if I leave at 6 a.m., I could be in before 7.
Wow.
But most of the time, it's an hour and a half.
That's a schlep.
It's a schlep both ways. 3 hours of my day is shot.
Yeah.
It's really shitty. That's why I listen to podcasts. At least I can hear something. And that's how I got interested in podcasting, right? So if I hadn't had this commute, maybe I never would have done any of these podcasts.
Well, it's an upside.
Yeah, but
it is.
I have to.
I say that because I didn't have to drive that commute.
Yeah, I wish I had a smaller commute. I'm hoping.
Well, you're always welcome to come over to Drew and do things.
That'd be 20 minutes, right?
Yeah.
I don't know when that's going to happen. However, today we do have some cool virology for you. And if you like hearing about it, please consider supporting us, microbe.tv/contribute. We'd love if you gave $1 a month. You can go to that website and find out more about that later. And don't forget, there's a meeting down in Queenstown, New Zealand, the 2nd through the 5th of December of this year, the Australasian Virology Society. It's their 10th biennial meeting. You can go to avs.org.au for more information and to register. We have a bit of interesting news this week about 2 Ebola treatments that have been shown to be very effective in the ongoing Congo epidemic.
Yeah.
Just made the news, right?
Oh, sure did.
Yes.
This is a big deal. They decided to stop the clinical trials because these drugs were working so well.
They were testing a number of different therapies. Yeah, and 2 out of the, they were doing 4 different therapies, and 2 out of the 4.
Right.
They were doing 4.
2 out of the 4 worked. So 3 monoclonals and 1 antiviral. And 2 of the monoclonals did really well. One of them called REGN-EB3, which is made by Regeneron. It's a cocktail of 3 monoclonal antibodies. And the other one is called MAB114, single antibody. It's derived from an antibody from the blood of a person who survived Ebola in 1995. And the NIH developed that into a monoclonal. It obviously cloned out the genes encoding it and produced the protein. So it no longer has anything to do with that survivor. And these 2 worked really well. They like 90%, over 90% of the people who got the drug shortly after infection survived.
Which is like the inverse of the survival rate if you get no treatment.
Yeah, exactly.
53% of patients who got the drug died, and 49% who received ZMapp, Everybody remember ZMapp?
Yeah.
Mm-hmm.
ZMapp was the hero in the last West African epidemic. It was made by a small biotech company in San Diego. 3 monoclonal antibodies. And it didn't work so well this time.
So I read something that the dosing was different. So ZMapp was dosed lower than the other 2 monoclonals. And it's harder to make, and so they weren't able to make enough to use that same higher dose.
Okay.
Well, I guess that's going to be a big problem because it's gone.
Yeah.
Unless they want to try it in another outbreak. It's too bad. Of course, Regeneron is a big company up in Tarrytown, New York.
Yep.
Which is well known for taking many Columbia people.
Wasn't it founded by Columbia people? Or just they took a lot of people?
They take a lot of people. I just know there was a steady stream of people hired out of Columbia.
Yeah.
And, of course, the director of science, George Yankopoulos, was an MD, PhD student here. Did you know him?
I remember him. Yeah, I overlapped a little bit with him when I was in grad school.
Well, now he's a billionaire and you're not.
Yeah, and I am definitely not. Yeah.
So these are interesting. The Regeneron is a cocktail of 3 monoclonals, and so they have different properties. One of them has effector function, but it doesn't bind the glycoprotein. So effector meaning it could do, it could help get phagocytosed, whatever it's bound to, or do lysis.
Right, or block binding to the next cell to block infection.
I don't know how it would do that. It's got no glycoprotein binding. So where is it binding?
That I wasn't sure about.
It's got to bind somewhere on the virus particle, right? Because it's got to lead to these effector functions.
Right.
Then there's another one which binds the glycoprotein but is not neutralizing. So neutralizing is our classic viral assay. You add antibody to a virus, you do a plaque assay or some kind of infectivity assay, and it decreases the infectivity. So that's what we mean by neutralizing. So 2 of the monoclonals do not neutralize virus infectivity, but the second one has effector function, and it also binds the glycoprotein. And then the third one neutralizes infectivity. There's no immune effector function and no glycoprotein binding. How about that, right? That's reference 113.
Yeah, so I'm not really clear on exactly how these are all working, but it's nice to see they are.
I wonder if I went to this...
it's hard to imagine some of that.
Maybe they talk about it in the paper. Is that possible?
It is possible. I haven't dug into the references on it.
Let me go look right now. I'm going to go to PubMed, and I'm going to type in the first 6 letters of the paper. Here we are, Safety, Pharmacokinetics, and Immunogenicity of 3 Human Monoclonals. This was published in Lancet Infectious Diseases last August, a year ago. This was a phase 1 trial.
That's not going to get us at mechanism, probably.
Probably not. Let's go to the paper and see if they have bolded it for us now. Probably not. Introduction, evidence. No, we don't want that. Glycoprotein. Each antibody targets a separate epitope on the viral glycoprotein, and all 3 can bind the glycoprotein simultaneously. And that's in contrast to what I just told you.
Yes.
Yeah,
so they are glycoprotein binding.
Yeah, so the thing is, in this paper, there's no soluble glycoprotein binding because, Ebola infected cells make a membrane-bound and a soluble glycoprotein. So these monoclonals differ in their ability to bind the soluble glycoprotein, but apparently they all bind the membrane-bound glycoprotein at different epitopes.
Okay, that makes more sense.
Makes a lot of sense.
Definitely makes a lot more sense.
And 2 of them are neutralizing and one has just effector functions.
Okay.
Yeah, this is it. The 3 antibodies were selected to have the following complementary biological properties. 1 combines in vitro neutralization and effector function without binding to secreted glycoprotein. The second has no neutralizing activity but has a potent effector function and binds to secreted. And the third has potent neutralization but does not have effector function and does not bind secreted. So it doesn't, they all bind the membrane-bound glycoprotein.
Right. Okay.
So they're binding 3 different epitopes on the viral glycoprotein. And earlier this week, Alan had mused, I wonder if they could be combined.
Yep.
It would depend on the epitope recognized by the AB114.
Right.
Mm-hmm.
I wonder if we can find out. Since that's been developed by NIH, we can definitely find information about that. What are the chances of typing MAB114 into PubMed gives me anything? Yeah, there we go. We got 8 papers. Safety and tolerability. Which one should I choose? Structure and molecular basis for Ebola neutralization by protective human antibodies. Here we go.
That's probably it.
So 2 monoclonals. So there were 2 originally, 100 and 114, which recognize different epitopes. So I don't know if they're different from the the Regeneron ones. I'd have to look.
They might just compete with each other.
It would take longer than we have here today, but it's possible. Although you'd have to do another trial if you wanted to combine them.
Yes. And I was wondering, I know the antiviral compound didn't do that well in this trial, but I was wondering if maybe it in combination with one of these antibody therapies.
Yeah, although this
that would make sense.
But
this Regeneron one is really good.
Yeah, I mean 90% is pretty darn good for this virus, and as we've said many times before, this is a virus that should not really be causing outbreaks in a properly functioning world. So,
a properly functioning world. Our world is becoming less and less functional, apparently.
Yeah, you should see, it's totally expected that you would see an occasional case or two. And countries in the area where that's true, where they've got the reservoirs for it, you'd expect to have drugs like these on hand, and that'd be great. You could save 90% of those lives, but it would be 1 or 2 people. And, but these multi-thousand person outbreaks are a product of chaos.
So remdesivir is made by Gilead, and it is a nucleotide analog.
Right.
Specifically developed for Ebola and Marburg. All right, so now let's say we have these monoclonals, and if there's a future outbreak, you can immediately give them to the healthcare workers, right?
Right.
So I'd like to know how long they last. Do we know?
I don't
last
think we know.
In the bloodstream, you mean?
Yeah, and when
giving them prophylactically, and would you give them? Yeah.
How often do you have to give it? Because so let's go back to the phase 1 pharmacokinetics. Yes, that should tell us, right?
Yes, and are they human antibodies, or are they sort of chimeric antibodies with another species?
All right. First question I'll answer is the half-life of each was 21 to 27 days.
Oh.
So probably every 2 months you're going to need some, right? Now, that's often. Yeah. And these have to be given intravenously, so this is not trivial. It's a little harder than a vaccine, which you could just put in the muscle, right?
Right.
So, but for healthcare workers, that's probably okay.
That's doable. Be on the safe side, give a monthly injection of this stuff.
Although, we do have this vaccine, and all the healthcare workers are getting that, so I would guess they're going to get that, and if they happen to get sick, then they can give them the antiviral, and it'll save their life.
Mm-hmm.
Yeah.
And then as people are brought in who are already sick and they haven't been vaccinated, they could get these monoclonals, right?
Mm-hmm.
This all assumes, of course, that you can do this, because our understanding is that in this country it's difficult to treat people, right?
It is. I think that this news might help that a little bit.
I hope.
Because some people might not have been going into a treatment center because they weren't sure that there was something that could be done for them. Yeah. And now, if there's news that says, well, we have a new treatment that can help 90% of patients, that may be a big help.
So then the other question you ask, are these chimeric or humanized, right?
Mm-hmm. Yep.
I guess it's one of those two, because they're probably originally made in mice, right? And then either the antibody, the combining site is built into a human framework, right? Right.
The one that came from the survivor might be fully humanized. That would just influence how frequently you could give these antibodies before you developed an immune response to the antibody itself.
Right.
Yeah. So the ZMapp is chimeric. Say what it is in this paper, so I don't know. So
yeah, you might actually not want to give these prophylactically.
Yeah.
Why is that?
Because if you do generate an immune response against them, they're not going to work at all when you need them.
So
exactly.
You would make an immune response against the combining site? Is that what?
Maybe the combining site, or if it was a chimeric antibody, the FC region, the tail, for example, could be mouse.
Right.
And you could make a response.
You could humanize that, though, right?
Yeah. So my question was whether they'd done that or not.
I bet they did, because the let's look it up. So if we're going to get an answer to this, we can put in the name of the antibodies, and then I think Google would know this. So now I'm not getting the original.
They've probably humanized it if it's persisting in the bloodstream for a month.
Right. So it looks like on the clinical trials page that they list it as a human monoclonal.
Okay.
So can you make human monoclonals, pure human?
You can, with a lot of work in terms of genetics.
But I mean, so the thing about the mouse is you get a mouse hybridoma, right? And then you fuse it with your B cells from immunized mice, right?
Right.
So is there a human hybridoma equivalent?
There are mice that have part of the human heavy chain genes knocked in, or there are some cell lines that some people can do work with.
I see. So the NIH one, it was cloned from a human, so that's
right.
Easy.
I assume
that's
that
easy.
One probably.
Yeah. So they're probably chimerized. To what extent, we don't know. Okay. Anyway, that is pretty exciting. So it should, if you're coming into the treatment center and you're really sick, they can give you these. And now they ended the trial, and everyone's going to get these too, right?
Yeah.
Mm-hmm.
Who would need it? That's pretty cool.
That's the idea.
Are they going to have to pay for them?
Who the patients?
Yeah.
No, they can't afford it.
Right,
no,
that's what I mean.
Right. No, this is all coming out of international aid funding, I think.
Now this is going to be paid. The company will be paid, of course, right?
Yes.
Regeneron will be nicely compensated. The NIH,
they'll be paid by the WHO and CDC and the other.
And NIH will come out of their own budget. They'll take away my grant to pay for it, probably. Which will be fine. That will come out of their own budget. But, and that will probably be the case, since all the outbreaks are in Africa, that will probably always be the case, that they'll be paid by WHO and UNAID and all that stuff, right?
I hope so.
Yeah.
Unless they're occurring in an African country that can afford them. Yeah.
Right. Now, monoclonals are not cheap, I guess, right?
No, not cheap, not easy to manufacture.
Because I know the ones for respiratory syncytial virus are enormously expensive.
Right.
Mm-hmm.
And as biologics, they're very hard to make generic versions of, at least under current regulations.
Yeah, no, they are.
Anyway, there you go. We are stumbling through this. Hopefully you've learned. I'm still wishing I could find how these things were made. But, it's funny. I type in the name of the antibody and I get one article. Is that what I used originally for the search term? It's too bad there's no history in PubMed,
Yeah, I think we were just going to talk briefly about the fact that these drugs worked, and then we got interested in the mechanistic aspect.
I blame it on Brianne. She's the one who asked.
None of us had done our homework on that. So.
Yeah, I figured there were the Regenerons were humanized, so you could humanize everything but the combining site, right, Brianne?
Right. Yes.
And then if you put that in, you're still going to get a little response against the combining site, right?
You could get a little bit of a response against the combining site.
I know that people at Regeneron once told me they can do all kinds of modifications of these antibodies. In addition to chimerizing, they can pagylate them and do other things to really extend their half-life in the blood beyond these 27 days. So it's quite an interesting science. Okay, so that's our news for this week. We have a snippet for you, and I picked a very old paper. Well, maybe it's not so old.
12 years ago is definitely older than the norm for TWiV.
For us.
It is not the oldest paper we've covered, though.
No, 2006 is not really 2007, January 2007. It's still in the 2000s, right? But
it is, yes.
It is.
It seems, I know for a lot of people, it seems like an old paper. They're used to us doing current literature, but I thought
it is pre-TWiV.
Yes.
2006 is pre-TWiV. That's right. And so I thought we could go back to some older papers, and this one is cool. It's called A Virus and a Fungus in a Plant, Three-Way Symbiosis Required for Thermal Tolerance. And this comes from Marquez, Redmond, Rodriguez, and Rusink, and it was published in Science in January 2007. And I mean, already you can see it's old because it only has 4 authors.
Yes.
Yeah.
Whereas nowadays, papers have many. And this is kind of the early days of understanding all about beneficial effects of viruses.
Mm-hmm. This is the story I always tell in my classes, but I'd never actually read the original paper.
Exactly. We tell this. I tell it in my classes. People use it as an example. At Rutgers, you remember, Brianne?
Yes.
Siobhan brought it up, and I thought we should do this paper because it's really short and easy. It's a great snippet, and everyone would know from now on what we're talking about.
Yes, and in fact, in the introduction, they even mention one of the viruses we talked about at Rutgers.
That's right, the hypovirus that attenuates the virulence of the chestnut blight fungus, right? Is that what you're referring to?
That's exactly it.
Yeah, so this idea that viruses can attenuate virulence of other pathogens is known. So the chestnut blight fungus is Cryphonectria parasitica. Wiped out chestnut trees, all throughout the U.S.?
Yep.
Yes.
Came from, what the guy told us at the Rutgers TWiV.
He did.
I can't remember. That's going to go up
listeners,
next.
listen for next week.
Yeah, next week it'll be posted. We had a great conversation at Rutgers, and he talked all about where it came from, and it turned out that some trees weren't dying, and the reason is, which Don Nuss and others figured out, is there was a virus in it that reduces the virulence. So this story, though, is about a virus-fungal plant symbiosis, and it has to do with a grass called panic grass, which is a wonderful name. And the way I think of it is when the grass sees the lawnmower, it starts to panic. Actually, this has nothing to do with lawnmowers. This is a grass that grows in high soil temperatures like Yellowstone National Park, where these root zones are really hot. I mean, I think it's like 50 Celsius out in the park, and in the lab you can go up to 65 Celsius. And this panic grass, which is called Dicanthelium lanuginosum, can grow. And the reason has been known for a while. It's got a fungus in it, Curvularia protuberata. And I can say these because I've been saying them for 12 years or 10 years in my course. That's the only reason, because in the early days I used to mess it up all the time.
And what's interesting is that neither the plant nor the fungus can grow above 38 degrees by themselves.
By themselves, right.
But only when they're together, they can go up to 65.
It's a true symbiosis. They really need each other.
Yeah.
And so this is a thermotolerance story. And she points out there are also stories of drought tolerance, which is also conferred in some cases by viruses as well. So this was Marilyn Rusink was the first person to really push this forward with viruses and plants, and has continued to do it. And so they said, okay, we know about the hypovirus that reduces the virulence of the chestnut blight fungus. Could there be a virus in this fungus in the grass that makes the combination heat tolerant? So they simply extracted RNA from the fungal cells, and they found a double-stranded RNA virus. Very simple.
It is just simple as that.
They said, we're using a protocol for nucleic acid extraction, enriching for double-stranded RNA. They ran it out in a gel, stained it with ethidium bromide. These are all
There
old
it is.
techniques. There it is, which most people don't do anymore, I guess. Maybe in plants they do, and in fungi they do, but nowadays you'd be doing it differently. And there's two RNA segments.
Nowadays, you'd probably be doing a much more complicated experiment that wouldn't be necessary.
Yeah.
I'm
and wouldn't be as clean.
I'm kind of sad about that. Been reminiscing a lot about that lately, that we used to do pretty simple experiments because that was the technology we had, right?
Mm-hmm.
And now we have incredible tech, and people drill really deep into the details, but often don't have the big picture. We did a paper yesterday on TWiV where they took all the microbiomes, they took many microbiomes of 4 different human sites, and they plowed through the data, and they found 500,000 open reading frames less than 50 amino acids long, okay? Belonging to 4,000 different protein families, something like that. And this is really cool, but you know what? What the hell are you going to do with that? What are you going to do with 500,000? There's not enough NIH grant money to even
There aren't
start.
enough graduate students in the world.
No.
It's kind of sad that, people do all these screens like that all the time, and most of it never gets followed up. It's kind of sad. So this kind of paper here where they extract the RNA, run it out in the gel. Hey, look, Marilyn,
there's a virus.
There's RNA here. You get all excited. And so this is an RNA. They sequence the 2 RNAs. One encodes RNA polymerase. The other one has open reading frames. They don't know what they encode. They can see particles. They purify the particles from the fungus, 27 nanometer particles. And the virus is transmitted vertically in the conidiaspores. So they call it Curvularia thermal tolerance virus. CTHTV.
You're watching CTHTV.
Yeah, exactly. Exactly. Actually, you're listening to Microbe TV right now.
Yes.
So they show now in a series of interesting experiments that the presence of this virus is needed for thermal tolerance. So the first thing they do is get rid of the virus.
Right. Let's cure the virus.
So they get some fungal colonies that have a low titer, and then they do some desiccation and freeze thawing. This is low tech. This is what I love, right? Low tech. And they get rid of these disruptive virus particles, and eventually they can get a subculture that doesn't have any virus in it. You can extract RNA, you don't see it. You can do PCR, electron microscopy. No virus is present. So that's really nice. That's what you want to do, take the virus out. And they show that if you try and grow plants with the fungus without the virus, they won't grow at high temperatures.
They're pictures of dead plants.
Yes,
I
they
like
look
it.
sad.
Live and dead plants.
Yeah, green and yellow and shriveled. Dead or chlorotic as that's the right word for a yellowing plant and healthy plants. So the virus is needed. And then, listeners of TWiV, what would you do next? You remove the virus. The plant won't grow at high temperatures. What would you do next? You put the virus back in. And so they have fungi with the virus and fungi without, and they let them fuse, right, on plates. Hyphal fusion or anastomosis, and they transfer the virus to the other fungus, and that one confers thermal tolerance.
Yep.
Mm-hmm.
So you find a virus, you take it out, and you do the phenotype, you put it in, you show the same phenotype.
Yes, Bio 362, pay attention.
Now the thing is, this is cool. They say, could we confer heat tolerance to another plant?
Right.
Like a tomato. So, and of course, tomato, I guess, would be cool because if you're growing them outside and it gets hot, you wouldn't lose a lot of them, right? That would be kind of interesting. So they put...
Also, tomatoes are easy to grow in greenhouses, so they're a good system.
Yeah. So they do show that they can get some tolerance, but it's not 100. They can't get 100% fungal colonization of the tomatoes, so they still get dead tomato plants. But it seems to work to a certain extent. All right.
The tomato plants certainly look better in their pictures when they have the virus.
Right. Yeah. The ones without, they're really they're brown and shriveled.
Mm-hmm.
They call them, yeah, they're dead plants. So it can work in, at least in tomato, right?
Mm-hmm.
So a grass is a monocot, right?
Yes.
And
yes.
so tomatoes are eudicot?
Yeah.
Why is it U?
It's a true dicot, I guess. That's what the U
Eudicot.
usually means, but I don't know what the false dicots are called then, if there are.
Most dicots have a pollen structure that differs from monocots, and a minority of dicots. This large subgroup of dicots is called eudicots.
Okay.
So this is a tripartite symbiosis. And a virus, a fungus, and either a monocot or a eudicot plant. And the fungus is an endophyte, of course, because it's living inside the plant. And they say complex tripartite symbioses have been found among arthropods, bacteria, and mutualistic bacteriophages. And that is it. This is basically 2 pages.
Mm-hmm.
And a really cool finding by 4 people. And I kind of yearn for that kind of science still.
Yes.
I'm not sure you can do that anymore. I don't think you can do small science like this.
And clear figures that are sort of easy to make out.
Yeah.
Now,
there is a correction, but it's a pretty simple one.
Yes.
There's not to say that we shouldn't do complicated science, and it's all, I definitely think that's fine. We can do screens, we can do all kinds of metagenomics, but there should also be a place for small science as well. And I'm not sure we can do that. I'm reading a biography, a book about Nobel Prizes, and there's a chapter on Andrei Lvov, who got a Nobel Prize, along with Jacob and Monod in 1965. And Lvov, of course, discovered lysogeny. Pretty important thing.
Mm-hmm.
Right.
And you know why he discovered it? He didn't like working with populations of cells because he didn't want to do statistics. So he worked with single cells of bacteria. And he noticed that they kind of lysed randomly, not all at once, because he didn't like statistics. I love it. Anyway, he liked to work in a small group, him, his wife, and a technician. That's what he did. And he worked in the lab. And that's how he got most of his results. And I think people should be able to do that. But it's not the way of science anymore.
Yeah.
You know, although I would argue that you could still do good science in a small group like that. Right. But it's not happening.
That's what those of us at small labs try to do.
Yeah. But it's hard, as you know.
Oh, yes.
It's very hard to do. And I could not do it here at Columbia. It's very difficult. And at other places. Okay, so now we have a paper for you, which is one of our favorite subjects, bats. And here we have more authors. This is a Nature Microbiology paper, which came out in May. And Brianne and I at ASV heard the lead author talk about this, Linfa Wang. Dampened NLRP3-mediated inflammation in bats and implications for a special viral reservoir host. And this comes out mainly places at the Duke National University of Singapore Medical School, which is in Singapore, and the Chinese Academy of Sciences, a variety of different locations. And the first author is Mate An. And the last author is Linfa Wang. Bats. And you remember Linfa's talk at ASV?
It was a great talk. In preparing for this TWiV, I actually pulled up my notes from that talk. So
there's a wonderful commentary on this article in the same journal. And it starts off with, what is interesting about bats?
Mm-hmm.
Yes.
They're only mammals that can fly. They say truly fly.
Right. As distinguished from flying squirrels, which glide.
They glide, right? They don't flap their...
Or they're falling with style.
Falling with style. And of course, they host a lot. For us virologists, we are interested in because they have a lot of viruses in them. And some of those viruses make us sick. And Linfa made this interesting diversion, or for a few minutes, he said he always thought bats had disproportionately more viruses than anybody else. And he had a bet with someone else, Peter Daszak, who said, no, that's not true. And Linfo said it's proven now that they have more viruses. Yes. But the author of this article says questions remain whether bats are disproportionate hosts. So this author, who is David Hayman, Hayman, thinks that it's not settled. Maybe he needs to talk to Linfa. Right.
I think that came out of a line from a recent paper from the Daszak lab. Yeah.
Anyway, bats have a lot of viruses. And the other thing that's cool about bats is they age well.
Right.
Right? And
so the commentary article has these nice graphs of things like body mass versus life expectancy, and bats are just off the curve on most of them because for a creature that's smaller than a mouse, they live several times longer than a mouse, and their metabolic rate is really high, so they should have short lives, but they don't. Right. They're just anomalous in multiple ways.
Yeah. And so one of the ideas is that their ability to age correlates in some way with their ability to tolerate viral infections. And we've talked about this a little bit before on TWiV. We've had a couple of TWiVs. One I remember a long time ago, we talked about the first bat genome sequences, where it looked like innate immune genes were duplicated and triplicated and so forth. And the idea maybe being that, in flight, you have a lot of oxidative radicals made and you need to deal with them. And so maybe you have mechanisms that have evolved, and those also deal with virus infections.
Right. The idea was in those papers is that there's increased DNA damage responses and decreased inflammation because you're not responding to the damaged DNA.
Right. The other thing that I found interesting is that some innate immune, some interferon genes are on constitutively in bats.
Yep.
Right.
And he says in this article, this constitutive activation is hypothesized to reduce the need for affinity maturation to make high affinity antibodies. That's very interesting.
It is. That was all new to me. He'd mentioned the other information about the higher levels of interferon in this paper and in the talk, but I hadn't heard about the other.
Yeah, so the idea would be you have a lot of interferon, and that deals with a virus infection before you need to make antibodies, right?
Mm-hmm.
Right.
So, and then this is kind of curious because they say maybe this has something to do why some bad viruses are pathogenic in other hosts. They get into a host where there's not as good as an innate response, and they go crazy. And I don't know if that's true or not, but
right. So I guess that all kind of fits into the flight as fever theory.
All right, So explain flight as fever, yes.
Yeah, so bats have very high metabolic rates for mammals, and they're more consistent with birds in that respect. And it's because they have to fly, which uses huge amounts of energy. So they have really, really fast heart rates and all this going on metabolically when they're flying, and they have high body temperatures. And the theory has been that is like an ongoing or recurrent fever. And so the bats are able to either control or tolerate viral infections because they're always running a fever, effectively.
Mm-hmm.
They run at such a high temperature that the viruses can't really get a foothold. The contrasting theory, which really kind of addresses more of the aging aspect of it, is that in fact they have tamped down inflammation and their immune responses, and that would explain things like the slower aging, because you don't have the inflammatory processes going on. So it's almost like, they're running at a high temperature, but they're not acting like they have an inflammation. They're metabolically, they're tamping down inflammation. And so these are two theories for how bats deal with their high viral loads.
Right.
And as Brianne mentioned, there's the STING pathway, which detects DNA. Seems to be dampened. I think we talked about that a long time ago on TWiV.
Yeah,
we did
as well. And today,
episode 484.
And today we will talk about another inflammatory pathway. That's the subject of this paper. By the way, a paper just came out showing Sting is now playing role in RNA virus infections. So we are always surprised. Okay, so this paper has to do with NLRP3.
Yes.
Nucleotide binding domain and leucine-rich repeat containing protein.
Yes. So that's what the NLR part of this stands for. And then the NLRs are a big group of proteins that seem to sense different types of cell stress or microbial products. The P tells you one of the types of domain that is in the protein, and then the 3 is for which number that has that domain. The P is for a pyrin domain.
Right.
Is that like the stuff you cook in that doesn't break?
No, that's Pyrex.
Oh, it's Pyrex. Okay. So these are responding in some way to various stresses, including viral and bacterial infections, right?
Yes.
And what's the response? What do they do when they sense these things?
So they will lead to the production of a protein complex called the inflammasome.
Mm-hmm.
Which has a much simpler name than NLRP3.
Yes. Which some of my colleagues like to refer to as the pinwheel of death. Because it does look just like a pinwheel.
Yep.
That activates a caspase and subsequently activates some cytokines like IL-1 beta. Mm-hmm. It also can lead to a type of cell death called pyroptosis.
Mm-hmm. So this can all be bad, right? It can cause immunopathology.
Well, this is an inflammatory response,
right? Which is meant to deal with whatever the insult is, but which
right.
can also have immunopathologic consequences.
Sure. Right. So NLRP3 and the inflammasome I think of as the fuse, and the virus gets in and it trips this and lights it, and then you've got the inflammatory response because this all goes off.
So, Alan, is it a screw-in fuse or a snap-in fuse?
I'm not sure. No, it's more like a fuse that gets lit.
I bet. Yeah, I bet nobody knows what a screw-in fuse is anymore. I used to have those when I was a kid. I was so afraid to screw them in. No, we
looked at a house that had fuses in a fuse box when we were shopping.
Fuse houses. Boxes now they have switches, but they used to have fuses.
Right. Oh yeah, they're breaker boxes now.
And they used to have to screw the fuse in. And
right,
when
like
you
a light bulb.
Sometimes when you put a new one in and it would connect, it would make a little spark.
Hmm.
It's scary. It's 110 volts. But this
doesn't sound fun.
But this is not the same.
No, different kind of fuse. This is more like a fuse and a stick of dynamite.
So their hypothesis is that this NLRP3 inflammasome, it's a key player in infection, as we said. It also has an effect on aging. We didn't mention that, right? But but this inflammation is linked to aging and age-related chronic diseases, right?
Yep.
They say it differs between bats and other mammals. That's the idea.
Yeah. One thing that's sort of interesting about NLRP3 compared to a lot of the NLRs is that NLRP3 seems to respond to a really broad array of stimuli. Mm-hmm. Maybe as if there's actually some other receptor upstream or some other thing that's binding, because most types of cell stress tend to turn on NLRP3. So they use a whole bunch of different types of stimuli here to hit NLRP3. And that's one sort of question in the field is what actually does NLRP3 bind? So as people are listening to this, they may get confused about why there are so many different stimuli. But NLRP3 is really nice because it does seem to broadly bind to cell stress, something that has to do with cell stress, and might be really the first one to look for with viral infection.
So when we give exams to our students, do you think we're activating?
They activate NLRP3, right?
Yeah.
Absolutely.
So if we did some kind of readout, we could measure it, right?
Probably.
Could measure IL-1 beta, right?
Right.
Possibly, yes.
So next time you give an exam, you could ask them if they'd like to donate some serum for that. So then the series of experiments are to compare the NLRP3 in bat and non-bat cells. So they have bat primary cells, immune cells in particular. And so when there are two signals involved in this, as Brianne alluded to. There's a priming and an activation signal of NLRP3. And then when you get both, you get these specs called ASC specs. It's basically the assembly of a protein which has the name ASC, apoptosis-associated spec-like protein containing a card domain, which is another kind of protein domain, ASC. And you can see these specs in the cytosol, right, with antibodies.
Yeah, and for people who are uncertain about what sort of is meant by the priming versus activation, really priming just means that you transcribe all of the proteins needed for this process.
So you make the proteins and then somehow they're activated, right?
Exactly. Yep.
Okay. And then these specs recruit and activate caspase-1, and then you get, as you said, pyroptosis, inflammatory cell death, and get production of IL-1 beta, which is made by cleaving a precursor, right?
Exactly.
So there are a number of ways you can measure this activation of NLRP3. So they do this in peripheral blood mononuclear cells from bats. And they start with one species, Pteropus electo. Sounds like a Harry Potter thing, electo. Right? And then human, or they also check bone marrow-derived macrophages or dendritic cells of bats and mice. Now, Brianne, they add LPS, which is one of the that would be a primer.
That's a priming signal. So that leads to transcription of IL-1 beta and NLRP3, and then they also add a second activation signal.
Yeah, ATP, for example. Now, something else is sensing LPS, right?
Right. That's some type of toll-like receptor, probably toll-like receptor 4.
And then there must...
And LPS is lipopolysaccharide. It's a bacterial component that triggers off inflammation.
Yes.
So it's used a lot in these sorts of things.
And you can buy all this stuff pretty much.
Yeah.
You can.
Off-the-shelf chemicals. You can't buy it at Whole Foods, but you can buy it at Sigma or Fisher or whatever.
Yeah.
And that's why science is expensive.
Yes.
All right, so then they look for specs,
right?
Basically with an antibody against one of the proteins in the spec. And then they can use fancy microscopy to quantify, which we don't need to get into. They can do flow cytometry, which is now why they have 15,000 authors, or a lot of authors on the paper, because everyone is doing something different, right?
Right.
So you treat human cells, you get specs, they can quantify it. In bat cells, you get low levels of specs.
Very low levels.
Right. So lower specs in bat cells. They measured pyroptosis by looking at IL-1 beta.
And also release of the cytosolic contents outside of the cell.
Right.
Right. So they're seeing if the inflammation pathway kicks off and the cells lyse.
Exactly.
And all of this is lower in bat cells compared to human cells, right?
Right.
That's the bottom line. So there is clearly a difference right away in specs, IL-1 beta, cytosolic contents coming out, which would be from pyroptotic cell death, right? They're measuring an enzyme. Yeah,
they measure an enzyme, but the enzyme is representing cytosolic contents.
Yeah, normally it's in the cell, right?
Exactly.
Okay, so that's part one. Part two. So we don't have specs and we don't have this activation. So what's going on? Is it NLRP3, the adapter, or both? So NLRP3, as Brianne mentioned, is transcriptionally turned on, and it's via toll-like receptors which sense LPS, and NF-kappa B is involved as well. So they treat cells with TLR agonists of various sorts to hit different TLRs. And when you do that, you get pretty good NF-kappa B activation in bat cells, right?
Yeah, so NF-kappa B works in bat cells.
NF-kappa B works, but when you use the TLR stimuli, you don't get induction of NLRP3.
Right, and again, it's just NLRP3. All the other stimuli seem to be made
although it works in mouse cells of various sorts, but not in the bat cells.
Right.
And they look at the basal level of these mRNAs, and they're pretty much comparable. So it's not that there's a difference in basal levels, right? So there's something going on in the induction of NLRP3. The next thing they do is they start, since we don't know a lot about these proteins in bat cells, they look at the mRNAs and they find an interesting splice variant, which skips a particular exon, which hasn't been seen before. And this accounts for these transcripts missing this exon. You'll see in a moment why that's interesting. Over 60% of the mRNAs in different tissues of this particular bat. They have evidence that these isoforms are less functional in terms of these assays that we're talking about, spec formation and IL-1 beta cleavage, than the other splice variants. And so they have in total 4 isoforms. This is one of the 4. These all four transcripts seem to be dampened compared to human, and this alternative splicing seems to be a major part of that. ASC isn't dampened; it's just NLRP3.
Right. So it could make the specs if NLRP3 was acting at normal levels.
Right. So there's something with NLRP3, and a big part of it is missing an exon in one of the transcripts.
Right. So basically, they've put ASC from bats into human cells, and that ASC works just fine, but they put bat NLRP3 into human cells, and bat NLRP3 seems to be the problem in actually leading to spec formation and IL-1 beta.
Yeah, and this particular exon deleted one doesn't work at all. It works really poorly, and that's 60% of the transcript. So that could be a big part of the problem. Okay, next, they look at some other bats because, there are lots of bats, and we're just looking at one. You might miss something.
Could be an atypical bat, yeah.
Could be an atypical bat. So they look at ASCs and NLRP3s from other bats. And I just wanted to point out here they make reporter constructs where they fuse, ASC or NLRP3 to different proteins that can be assayed. And they have wonderful names like plum and citrine. There's not just red and green and yellow anymore. What is the one that I like? Tomato is a good one.
They're making me hungry.
What I thought was interesting here was I didn't realize that there are 2 major bat lineages, 2 suborders within the bats, and they're Yin Pterochiroptera and Yang Ochiroptera.
Oh, look at that! Yes,
yes,
very nice.
So those are the two halves of the bats.
So this exon deletion is found in other bats, in other tissues, right?
In both of the major lineages.
And they assess NLRP function in these different yin and yang bats.
Right.
And basically get the same results as with the others, right? Low activity of all the NLRP isoforms. And then they start making chimeras between the human and the bat proteins, right? NLRP3s, which is, you can shuffle things around. And they conclude that one part of the protein called the LRR, this leucine-rich repeat domain, I guess that is.
Yes. Yep.
Seems to be responsible for reduced function. So there are amino acid changes in that appear to be responsible for that. And that's interesting because that's the part that's missing in the exon 7 missing isoform of the RNA.
Right. And that's also interesting because that's the part of NLRs that is thought to bind to the ligand.
And what's the ligand?
Unknown for NLRP3.
Yes, I know. Okay. And so that domain is under positive selection in bats, which means it's changing over time. So it's responding to something. So this LRR is really the main player, it seems, in the dampening activity of the NLRP3 isoforms. So far we have NLRP3 function is reduced in bats compared to non-bats, and it seems to be because of changes in a very small part of the protein.
And this seems to be an ancient adaptation that probably goes back to the origin of bats.
Right.
Yeah.
So what happens if we would put viruses in here?
Well, that's a good question, because so far we've been putting chemicals that you can buy.
Right.
Albeit not cheaply, you can buy them and just go, so what does that mean? Yeah, this is all about viruses. So they do that. They put some viruses in here. So they have, let's describe the cells that they're using. They have they have NLRP3 knockout immortalized macrophages. From a mouse. So they're lacking NLRP3 gene, and then they put in human or bat NLRP3 with citrine. Citrine is a lovely word, isn't it? Yeah. So now they've put NLRP3 into human cells, and they're going to infect. I guess it's so they can infect them, right?
Yes, it is.
Otherwise, they could do this in bat cells.
Right, but then you'd have so many variables between the two different cell types.
Yeah. We know that the bat NLRP3 is what is causing the reduced response. So they infect these with influenza A virus, which is known to activate the NLRP3. And they say, well, the isoforms were less responsive to infection, so they didn't respond as much as human, but the viral titers are not affected by this absence of NLRP3 activation.
Right, and that's sort of the really interesting part.
That the viral titers are not affected, right?
Right. Even though there is a change in how much sort of this immune response is being made, there is no change in how much of the virus is actually being combated.
Yeah, you would think if this, if NLRP3 were making a big difference, you would see more virus, right?
Right.
But apparently, so this is in a human cell background, right?
Mm-hmm.
So...
No, this is in a mouse cell background here.
This is a mouse cell background, yeah. But it's still funny that, well, I guess NLRP3 is simply not doing anything. And you have to argue that whatever other systems are in place in these mouse cells, they're acting normally, right?
Right.
And then they do another experiment. They have bone marrow-derived macrophages from mouse or bat. They prime them with LPS, then they infect them with influenza virus. They measure spec formation. They measure IL-1 beta secretion. And again, the specs, the beta secretion, minimally or not induced in infected bat macrophages or DCs after infection with influenza virus. So again, these are mouse, right?
Mm-hmm. Yeah, these are all mouse.
And these are so this is kind of a control, right? Because these are all mouse. These are not the hybrid ones. These are freshly isolated.
Well, they're either freshly isolated from mouse or freshly isolated from bat bone marrow.
Oh, yeah, mouse or bat. So the bats are minimally induced, whereas the mouse are induced more, right?
Right.
And they try another virus. So this is, so far, influenza virus. I'm not sure it infects bats. So they try a bat-borne zoonotic reovirus with a double-stranded RNA genome. And that's kind of cool because our plant virus was a double-stranded RNA virus, right?
Mm-hmm. And this virus had never been shown to be sensed by NLRP3 before.
Mm-hmm. So they use this to infect their reconstituted IMAX, they call them. Hey, Apple would have some say in that IMAC, right?
They would.
Little I, big MAC. And decreased activation from the bat compared to human with this virus, with this bat virus. So that's cool because now we're looking at a bat virus, not a human virus.
Right.
Less inflammasome activation. And then they look at a third virus, which is interesting, MERS coronavirus, which infects people, but is thought to have at some point in the past originated in a bat via a camel, so bat-camel people, right? So they do the same thing. They have their the mouse cells are not permissive, so they have to infect human and bat PBMCs. So they can't use their IMAX on anymore, unfortunately. And I want to point out permissive means the virus will not replicate in them. Well, that's what it's supposed to mean. I don't know if they're actually using it in the correct way. So they use human and bat PBMCs. Mm-hmm. MERS corona infection activated the spec formation, the IL-1 beta secretion in human. It could be abolished by an inhibitor of NLRP3.
Right.
But it did not, the virus did not trigger this response in bat PBMCs. Although titers only differed minimally. So these cells do make virus, which is good, because if they didn't, then you wouldn't know if they're infecting them unless you did some other things. And much less NLRP3 and a much less inflammasome response in the bat PBMC. So that's a really nice experiment, I think.
Yeah.
Because that is pure human versus pure bat cells. And
right.
You can see the big difference. And that's it. That's the paper. So dampened transcriptional priming of NLRP3, a splice variant that messes up the function of the LRR domain. And there's a consequence. Whether you use chemicals or viruses, you get a dampened NLRP inflammasome production.
Right. So there's this ancient adaptation in bats that's fundamental to them being bats, apparently, that causes them to be to not trigger this inflammatory pathway that's triggered by viruses in other mammals.
Now, when bats arose, of course, there were already viruses.
Sure.
Right.
And so as they evolved to be able to do their flight, then they became, more resistant to viruses, I guess. Both things happened at the same time.
Well, not exactly more resistant, though, because just more tolerant.
More tolerant,
right.
Yes. Yeah, resistant to pathology.
It's not so much the flight as fever theory, because they're not clearing the viruses, they're not cooking them out. They are avoiding the inflammatory response that causes a lot of viral pathology, and so viruses that would make other mammals very sick don't make bats sick because they're not generating the inflammatory response, but they can carry high viral titers. Right.
They're sort of coexisting with the virus.
Right.
Here's my theory: the virus made them able to fly. You understand that, right? The virus,
yes.
They became adapted to not die from being infected and let the virus replicate.
And that gave them the metabolism that allowed them to tolerate flight.
Yes.
There you go.
And one day a bat went to the edge of the cliff and it was infected and took off. It didn't have to happen the other way. It could have happened. And viruses could have helped them acquire the mechanisms for flight. Who knows?
Sure.
Sure.
Let's see, a couple of things here in the discussion I wanted to point on. As Brianne pointed out earlier, no unified mechanisms for activating NLRP3 have been identified for viruses. That's what you said, right?
Yes, that is exactly what I said.
Right.
And it's not even just for viruses. All sorts of diverse things seem to activate NLRP3.
Activate being induced to transcription of, right?
Or actually, no, induce spec formation and IL-1 beta cleavage.
Mm-hmm. All right, and they have some candidates here that might be, but further studies are needed. Okay, so then they say observations in wild and experimentally infected bats suggest they tolerate disease even during transient phase of high viral loads, and that's been done with Ebola, MERS, coronavirus. You can get up to 10 to the 7th TCID50s per gram of lungs with MERS, not MERV. So that's something we were talking about earlier. And so maybe, the adaptations they've made for flight have something to do with that. Enhanced immune, so flight as fever hypothesis, which we talked about.
Mm-hmm.
So their results support an enhanced innate immune tolerance rather than an enhanced antiviral defense in bats.
Right.
I think that's probably reasonable conclusion. What do you think so far?
Yeah, I agree. And I think that there are a few other papers out there, including the Sting paper that we talked about, that sort of make it look like there is this global change in innate immune activation in bats, which is really cool.
Yeah, it's very cool. There's clearly a lot more to be done, obviously, because this is just one aspect. NLRP3 is one aspect, and you could look at lots of other things as well, right? And in fact, they go into that, nicely in the commentary.
They do.
What things are left to look at.
They do, and they talk also about how much of this you would love to do in sort of in vivo and see what changes we see in bats in nature, but obviously that would be really challenging to do.
Yeah.
And to know whether this is all viruses or just a subset of viruses that bats can tolerate.
Yeah, they make this interesting point. Are bats adapted to tolerate all infections or simply those they have co-evolved with?
Right.
They say rabies and related viruses persist, but they can kill bats. Are these the only exceptions? And they say the MERS results in this paper are important because it suggests that these responses are general. And so, but I think more bats and more viruses need to be looked at. Yeah. Anyway, that's cool. And, this is not trivial stuff because bat cells and cell lines and so forth are not easy to come by. In fact, bats are not easy to come by.
No, not at all.
So you have to, LINFA has established a group where they can do these experiments, and they're doing breeding as well. And it's important stuff and not easy to do, but pretty exciting.
I think every time I hear about details of the bat innate immune response, I just think that it's so cool and think I need to start a bat lab.
Yeah,
bat lab.
Maybe you need
exactly. And my students would sing that all day.
You could go to Singapore.
I could,
because I'm not sure how Drew would like you to have a bat lab.
Maybe it's just some cell lines.
Cell lines would be good. Yeah,
you
could do that. Cell lines would be fine, and you could probably get undergraduates interested in that very readily.
I'm sure that I could.
Yeah. All right.
But you have to culture the petri dishes flipped over.
Very good. Which we do for plaque assays anyway.
That's right. Yes.
Not everybody does that. It's interesting. I've always done it, and I contend that it makes the plaques better because you don't get the water dripping down on the agar, right? You did it when you were here.
Yeah. I never questioned why we did the plaque assays upside down, but yeah.
And people have told me they don't do it, and I say, well, good. I'm glad it works for you, but we're going to flip them.
Yeah.
Yeah.
All right, let's do a few emails.
Sure.
Arnow writes, thank you for your amazing podcast. On the last TWiV 555, you talked on RNA viral populations, and you sustained that quasi-species was a dated and not very appropriate term. I find it surprising as lots of current articles on viral populations use quasi-species, even if quasi-species applied to RNA viruses has been controversial, e.g., Esteban Domingo versus Eddie Holmes. I'm not aware of any decisive proof against the existence of viral quasi-species. Would you mind to clarify your position? It's not a matter of whether they exist or not. I just say that people don't like to use the term quasi-species anymore. So when I go to meetings, they use things like swarms, mutant swarms, or clouds, mutant clouds, right? Mm-hmm. Simply because quasi-species it has a weird connotation. First of all, there are no real species. I mean, we use species in viruses for nomenclature purposes, but there's no real species definition is very different from what you see. So to say quasi-species is even weirder. So that's why people are just trying to rename it. So I'm not saying that a quasi-species doesn't exist. Absolutely. And I love the word quasi-species, but other people are using different terms. And since I can't change very readily, I still use quasi-species because I think it's a cool word. But I just want you to know that you will see other things being used. Is that clear? I think so. I don't dispute that they exist. I'm just telling you that there's a different nomenclature. Okay, or now. Okay, Brianne, can you take the next one?
Sure. Caitlin writes, Dear Autonomous CollecTWiVe, after a 2-year Twix bender, I have finally caught up and am writing in to mark the sad and joyful day. I do not exaggerate when I say that your voices have been a constant in my life and have frequently helped me get through difficult times. Your podcast is truly a gift to the world. Unfortunately, I have absolutely nothing useful to add to the virological discourse. So instead, I list some facetious suggestions for special episodes that have occurred to me over the past couple of years. They may not be suitable for episode 600, but maybe for episode 666. First, an exercise in masochism. The TWiV team plays some anti-vaxxer or other fake science podcast and debunks it. Appropriate pauses for horrified screaming, furious rants, and loud expletives. Yeah, that I think there'd be a lot of rants going on there.
Yeah.
TWiV After Dark. The TWiV team tells their most shocking, terrifying, and unbelievable laboratory tales. Turn it into a contest. Which host will win? In the categories of scariest incident, strangest coworker, weirdest experiment, dumbest mistake, and favorite lab memory.
We'd get sued.
Yeah. Yeah, I've got some good entries, but I don't know if I can tell them.
Yeah.
Drunk TWiV. This should be fairly self-explanatory. For extra fun, it could be done through a drinking game. Dig out the old TWiV bingo card and have a host do a shot every time someone drops one of the bingo phrases.
Yeah. That one we could do in front of an audience.
Right.
That would be funny. The TWiV team attempts to decipher a paper that none of them have seen before, which is in a foreign language. They may use only their knowledge and Google Translate. Bonus points if it's in Italian for obvious reasons. Special guest, Andrew Wakefield, rubs hands together gleefully. Better yet, Rich bursts into his clinic unannounced, a VEC mic, and takes him to task on the spot. I doubt any of these TWiV episodes will ever exist outside of my imagination, but the thoughts made me giggle. The thoughts also made me giggle.
Yeah.
Finally, I want you to take this very seriously. F the haters.
Fork, I think.
Fork the haters, yes. If you get even one more letter whining about the weather segment, please forward it to me, and I will swiftly track down the ungrateful hooligan and insert a pineapple into their backside. TWiV is perfect the way it is. Caitlin in Seattle, where the weather is not nearly as rainy as I was led to believe.
Yeah.
Caitlin's funny.
Very funny.
Yes.
Now, I think Caitlin writes to TWiP occasionally, on which she is also very funny. These are great suggestions, and I can see that it's fun for you just to come up with them.
Yeah.
Yeah, those are good suggestions.
Since we couldn't figure out today if how the antibodies were humanized, and it was in English, I'm not sure how we would do on a foreign language.
Very
paper.
poorly.
We have to take every paragraph and translate it, basically.
Yeah.
Yeah. Fortunately, sometimes a lot of the science terms stay the same.
Right.
So some of the words would stay the same.
When was the last time you read or looked at a science paper in another language?
I do okay with papers on my topic of interest in French, but that's about it.
Where do you find them?
You can find them in PubMed.
Really?
Yeah.
I don't see any.
Yeah, stuff that comes up with the title in brackets is non-English, I think.
I see. We used to have an English a language requirement for a PhD. It was before either of you did your PhDs, but when I went to graduate school, it had just been lifted in our department. But before that, you had to take some foreign language as part of your PhD because, many papers were still in other languages.
Right.
And so German or French or Spanish or Italian, can you imagine as part of your PhD, you had to take language courses and pass them? Ugh. And in my program, they changed it to a statistics requirement. I'm not sure which one.
I'd have chosen language.
Yes, indeed. All right, let's do we have one more. Alan, take that one,
Sure,
please.
I'll take it. Chris writes, Dear TWiV team, in my opinion, if Zika virus infection causes microcephaly in humans, then it is very likely to cause other far less obvious outcomes in the fetus. The question is, how do we prove the association? The recent article published by Nielsen Sands et al. In Nature Medicine is a very interesting paper, certainly adding to our knowledge, but unfortunately not able to tell us much more about cause and effect. The lack of a control group does not help, but even if controls were available, it would only tell us so much. The paper describes a cohort of children born to mothers who had symptomatic PCR-proven Zika virus infection while pregnant. What we do not know, however, is whether Zika virus was transferred to the fetus in these cases. If only these children had been tested for the presence of anti-Zika IgM or tested by PCR shortly after birth. Antibody tests have had issues with cross-reactivity with related viruses, and therefore the results of antibody tests may not be as clear-cut as we would like. However, PCR tests would have been conclusive and would have indicated that the fetus had indeed been infected in utero. If one could have tested the newborns for evidence of infection with Zika virus, it would also be worth resting for evidence of other testing for evidence of other intrauterine infections, such as cytomegalovirus, toxoplasma, and rubella as well. However, even if infection of the fetus could have been demonstrated, it doesn't necessarily indicate that any damage results from the infection, although I believe it would be strongly suggestive. When a vaccine becomes available, the link between infections and pathologies should become much clearer. And what about subclinical Zika virus infections? Can they result in delayed childhood neurodevelopment and neurosensory alterations? I believe that the harder we look, the more we are likely to identify associations between Zika virus infection and pathology in newborns, children, and possibly adults, further justifying the need for an effective vaccine. Thanks for a wonderfully informative and entertaining podcast. And Chris is a senior lecturer in microbiology at Middlesex University in the UK. And I'd comment on two things. First of all, I would argue that PCR could not prove that they're active infection. It could only prove that material virus. But it would be, it's the test that you could actually do because you're virus from the fetus, most likely. So that is a reasonable and potentially very interesting test to look at. But the second thing is, I think some of the analyses, at least the epidemiology, have hinted that there are other outcomes besides microcephaly that are likely caused by Zika virus. And Vincent, you're probably more familiar with that.
Yeah, that was a paper we did on this. I don't know if you were with us that time.
I think I was, yeah.
Yeah, where they looked at this cohort, as he says, born to Zika-positive mothers.
Yeah.
And they found a certain incidence higher than, there were no controls, a certain incidence of...
higher than expected.
Expected in, learning and language and a few other things. And this is in the absence of microcephaly, right? And his points are good, There are lots of issues,
Absolutely.
But, that's a suggestive study, and I think it will stimulate some other studies as we go forward. But I think it was worth publishing with the caveats. And we talked about this, there's no control group. It's based on these standardized charts of many, many children, and it may not apply to that particular locale and so forth. But there are certainly hints of many more issues happening, yes, in children born to Zika virus infected mothers, and we need to have more information on it.
All good points.
Yeah. So these is all true. Absolutely. And thank you for pointing them out. Yeah. I think we also pointed out a lot of the shortcomings as well in that paper. All right. Time for some picks. Brianne, what do you have for us this week?
All right. I have a story that I came across on Twitter. It is about a student in the UK who is deaf and who went to school to study the sciences. And he was very frustrated because he had an interpreter during his lectures that was doing sign language, and there were no sign language terms for most of the technical words he was hearing in the lectures. So the interpreter had to spell all of the words out. And so he created the signs for about 100 different scientific terms to make sure that we have them and to increase the ability to do scientific communication.
That's pretty cool.
Yeah. And so apparently there weren't really signs for things like DNA or stem cell or things like that. And so he went through and created them all.
So when you do that, you have to make sure the signs aren't taken, I guess.
I believe so, yes. And these have all been sort of accepted into British Sign Language.
Which is cool. Abbreviated BSL, by the way.
Yes.
Yes.
This is BSL 1?
Right.
Yes.
I think he's a higher level of BSL. He's invented a whole... It's really awesome. I think that's great.
Yeah, I think that I hadn't first of all, I hadn't thought about how challenging that would be to have all of those terms spelled out.
Right.
And I think it's really fantastic that he went ahead and did something about it.
Yeah.
So I just looked this up because I don't know anything about it. There are over 50,000 signs worldwide. Not every sign language is the same. Wow. Mm-hmm. So I guess he had his particular sign language and he just made sure his science words weren't in it, right?
Right.
Right.
I mean, I can't imagine 50,000 ways to use your hands. Right? I mean, I know it's not just the hands. You do things with your body, right? And your head, that stuff. But I'm impressed.
Yeah.
It's amazing that he could do that.
Yeah.
Wow.
There's a little video with the article where he's being interviewed about it, and he demonstrates the stem cell sign.
Stem cell DNA, PCR, pipette, buffer, LPS. Did he do one for LPS?
I don't know about LPS.
So could he do the whole paper in sign language that we did
Maybe.
today? That would be cool.
Perhaps.
This is brilliant. I love it. Great. Alan, what do you have?
I have a little graphic design thing that I thought was really cool. It's called Show Your Stripes. And this is, you can put in any region or locality. You can put in your home state, and it will graph the temperature in a color-coded form from 1850 to 2018 for that place. So when you bring up the page, it comes up with the globe, and it goes from blue for cold into white, and then into red, and then into dark red over time. And it's just a really striking graphic representation of the dramatic increase in global temperature over that time period, especially in the most recent 50 years or so. And then you can put in your home state, or take it to your home state, or take it to anywhere you're interested in seeing the graph for, and you can download the image and stick it on stuff. I
just did New Jersey.
Mm-hmm.
You could see, every now and then there would be a red.
Yep.
But now they're mostly
just
all
red. Yeah.
Holy crap. How do you know, how many years is it all red? There's no scale, so you don't know what the years are.
Right. It's just a graphic. Yeah. So it's intended to give a quick impression of here's what's going on, and a localizable impression. In some places, there's not a whole lot of change.
And you can download it.
But most places, it's dramatic, and you can download the image.
And then you can put it on Twitter and say, this is the temperature for my state.
Page background, or post on Twitter.
Oh, this is so cool.
Yeah. I did both New Jersey and New York, and New Jersey seems much more dramatic.
Always the case, right?
Right.
Yes.
Oh my gosh, this is just... And, there are people who say this isn't real.
Right.
And now we're going to start having droughts and starvation globally because people don't want to do the right thing. They want to reverse all the regulations and so forth. It's so sad. People do amazing things and they do stupid things.
Yep.
It's just so sad. That's cool. All right. My pick is a article by David Baltimore called 60 Years of Discovery. And
this is cool.
This is a talk he gave at his birthday party last year, May 2018, his 80th birthday party. And he gave it at the last session of the day. It was a day full of talks, and then he sat in the front and gave this. And afterwards I said, you really should publish it. And he said, yeah, I plan to, and this is it. And it says, each of us is a story. Mine is a story about doing science for 60 years, and I am honored to be asked to tell it. And it's in the annual review of immunology, but, it starts with how he got interested in his virus phase, his immunology phase. And he always has written beautifully. So I don't know if you can get it. What do you think, Alan? Is it open?
Yeah, it actually is.
It looks like it.
So you just click on the link for full text from PubMed, and it takes you to full text.
Good. It's really, really beautiful. He's always, as I said, he's a great writer, and this is his life story, and it's cool. Check it out. We have a listener pick, which I've delayed for a couple of weeks, and I thought we could do it today. This is from Eric Delwart, our friend out in San Francisco. Dear Vincent and TWiVsters, I'd like to recommend a New York Times podcast by Dr. Robert Grant about pre-exposure prophylaxis, I guess, PrEP for control of HIV transmission. He asked the question, if we could stop the HIV epidemic, would we? Dr. Grant was first to demonstrate the efficacy of PrEP in preventing HIV transmission. He knows his stuff. He argues we now have a drug that effectively works like an HIV vaccine when consistently taken before HIV exposures. He clearly spells out the role of Big Pharma, in this case Gilead, in limiting access by charging $20,000 a year in the U.S. A powerful and touching testimony about a major problem translating scientific progress into actual public health benefits.
Thank you, Eric, for helping me with my syllabus for my HIV course.
There you go.
So he gives a link to the podcast, which I think is an episode of The Daily, which is by the way, it's the most popular podcast on the planet. It's a New York Times podcast. Have 1 to 2 million downloads per episode.
I think they're edging us out a little bit.
Just a bit. And then there is a link to about the patent. Gilead CEO insists federal government patent for HIV Truvada is invalid. Okay, I don't know what that's about. And then a summary from the CDC. Yeah, that's one I put there. Let's see what that has. Changes in HIV prophylaxis. It's just a very nice article on the effectiveness of PrEP. You know, it has a certain effectiveness on its own. It's even better with a condom. I think it's 70% on its own and 90% with a condom. I may be wrong because I can't find this is in French, so I can't find it right away. No, actually, it's in English. Just kidding. But so the drugs are very effective. So it's an interesting question. Why do you have to charge so much? If you charge less, you could get more people and prevent transmission, right? Any thoughts on this? It's the usual economics issues, right?
Yeah.
But why do they have to?
I mean, you've got one company with a monopoly on the market for this, and this is what they charge because they need to satisfy their shareholders, and that's who they're accountable to. And you can't really, I mean, you can hate the player or you can hate the game, but this is the situation.
So the Post article says the chief executive of Gilead defended the high cost of a drug, saying that its profits pay for continued research.
Right. That's the usual argument.
It said Gilead has spent $6 billion on HIV/AIDS research since 2000. So the drug costs between $1,600 and $2,000 a month in the U.S., compared with just a few dollars a month in some foreign countries. So it is cheaper elsewhere. Yeah. So what do you think? I mean, if it's cheaper elsewhere, that's good, right?
Sure.
That is good, but there are still people here who can't afford that.
Yeah, they say Gilead's high prices are preventing broader use of Truvada. Only 20% of the 1.1 million people who should be taking it are on the drug. I guess that's in the U.S. I don't know. This is where a company has to decide what it wants to do. You know, they could reduce the price, right?
Yeah.
Mm-hmm.
And get more people.
They're, I mean, as I say, they are accountable to their shareholders to turn a profit. And part of that is they're investing in research for future drugs, but part of that is also they're charging a lot of money for the drugs that they have in markets where they're allowed to charge a lot of money. Mm-hmm. And you could, I mean, we could argue that the problem is not so much the company. The problem is that we have a country that allows them to charge this price as opposed to other countries that don't allow them to charge that price. There's plenty of blame to go around here.
I'm wondering when the patent expires. Right, let's see, Truvada patent. Generic Truvada will be available next year.
Okay.
So that means
the other thing. The company is riding this monopoly to pay for their next, 10 failed clinical trials because drug development is expensive. And they've got a limited time to get that money because once something goes generic, the on-brand version of it just tanks.
Yeah, but they're also using it to pay for exorbitant CEO salaries.
Of course.
Mm-hmm.
And so 2020 is not far off. And so presumably other companies will make it generic, and that will help, right?
One hopes.
Now,
hopefully.
Question. When you make a generic, you have to go through a clinical trial of some kind, I presume, right?
It's an abbreviated one.
Abbreviated to show that your preparation is good?
We mentioned with biologics, it's different because the whole process of a biologic is what's certified. But with a small molecule traditional drug, basically you just show that it is equivalent chemically and pharmacokinetically. And then you get the go ahead to so there are tests in humans, but it's a drastically abbreviated because we already know this compound at this purity and this dose delivered to patients should do this, and you demonstrate that it does, and there you go, you're on the market. So it is a much shorter, cheaper route.
Okay, so hopefully more companies will get involved.
Yeah,
and maybe in a couple of years we'll have more.
And hopefully more people will know about Truvada.
Right.
I just want to also point out that Merck still gives away ivermectin for treatment of river blindness in Africa and other places.
Mm-hmm.
They don't get any money from it because that's what they decided to do. Of course, Merck is much bigger than Gilead, and maybe they can afford to do that. Maybe they're not losing that much money by giving it away.
Well, and they're also making money by charging large prices for other things that they make. So they're a profitable company.
Yeah. Always an issue. All right, that is TWiV 561. You can find it on any good and bad podcast player. The content is always good. The player may not be, but you can listen to it. And please subscribe so that you get every episode, whether you want it or not, and you don't have to listen to it. It's there, and then if you set your podcast player to only show the last 3 episodes, that works too. Or you could have all the episodes, whatever you want. But please subscribe. It lets us know how many people are listening. That really helps us in our efforts to fund the podcast. And if you want to help out in that respect, you could go to microbe.tv/contribute. We have ways either at Patreon or via PayPal, where you could sign up and give us $1 a month. And that would help us a lot to defray our costs and allow us to travel a little bit more and do remote podcasts, meaning outside of our usual Skype network here. And, of course, questions and comments, TWiV@microbe.tv. TWiV today, Brianne Barker at Drew University. Over on Twitter, she is BioProf Barker. Thanks, Brianne.
Thanks. It was great to be here. All of these papers were fascinating.
It's a nice collection. You bet. Go befriend a bat. Alan Dove's at turbidplaque.com. Over on Twitter, he is Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASV and ASM for their support of TWiV and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.